Hello, you're welcome to our lecture on biological techniques. Today's lecture will be on the preparation of microscope slides. Now, the most common slide preparation is called the wet mount slide and utilizes a flat slide and a cover slip. They want to now look at how to make a wet mount slide preparation. The first step in making a wet mount is that you place a drop of the sample in the middle of a clean slide. The sample could be water sample, it could be blood sample, it could be urine sample, or any other sample. You use a drop-in pipette to collect the sample, and then you place a drop on the middle of the slide. We are using a water sample, for example. Now, the drop-in pipette is used to collect the sample and is placed on the middle of the microscope slide. The next step, the second step now is that at an angle, place one side of the cover slip against the slide, making contact with outer edge of the liquid drop. So you place the cover slip at an angle at the tip of the liquid that has been dropped on the microscope slide. Now this is to illustrate what's been done. The cover slip is placed at an angle at the tip of the liquid that has been placed on the microscope slide. The third step now is that you should lower the cover slowly, avoiding air bubbles. The cover slip is then lowered slowly, and air bubbles are actually avoided so that they don't give a wrong result. So the cover slip is now placed, is now dropped, and it is now ready for viewing. Cover slip is now ready for viewing. The first step is now after placing it, you now allow the liquid to spread out between the two pieces of glass without applying pressure. Remove excess water and with a paper towel. Excess water is removed with a paper towel. The slide is then ready for viewing. Now, when you are viewing the organism, you now study and identify the object or the organism. You may make a drawing of the organism or object you saw. The field of view and depth of field increase as the magnification increases. By knowing the diameter, that's in millimeter, of the field of view for each objective length, the size of the object can be measured. Now, after viewing the object, usually drawings are expected to be made. You're expected to use a pencil, and when you make mistakes, you can erase the mistakes, but pencils are used for drawing. All drawings should include clear and proper labels and be large enough to view details. The drawings should be labeled with the specimen name and magnification. Labels should be written on the outer side of the circle. The circle indicates the viewing field as seen through the eyepiece. Specimens should be drawn to scale. That is, if your specimen takes up the whole viewing field, make sure your drawing reflects that. Now I want to look at one other aspect of microscopy, which is staining of microscope slides. The staining of microscope slides. Staining techniques can be employed to aid in the observation of cell parts. Two types of staining techniques are used. They are non-vital staining and the vital staining. The non-vital staining is the staining of dead cells or tissues, while the vital staining is the staining of live tissues. So the non-vital staining is used to stain dead cells or tissues, while the vital staining is used to stain cells that are still alive. The cells that are still alive are stained with the vital staining. Staining is a very complex subject but there are many simple staining techniques that can easily be performed. One of such staining techniques is called gram staining. So now let us look at the gram staining technique, how to stain a slide using the gram staining technique. I want to look at the gram staining technique. The gram staining technique is a common microbiological technique used to differentiate two large groups of bacteria based on their different cell wall constituents. The gram stain procedure distinguishes between gram-positive and gram-negative groups of coloring these cells red or by coloring these cells red or violet. Gram-positive bacteria stain violet due to the presence of a thick layer of peptidoglycan in their cell wall, which retains the crystal violet these cells are stained with. On the other hand, alternatively, gram-negative bacteria stain red, which is attributed to a thinner peptidoglycan wall which does not retain the crystal violet during the decoloration process. So the gram-positive bacteria actually stained violet, while the gram-negative bacteria at the end of the staining process are stained red. Now, what are the requirements for gram staining? The gram staining technique requires the following equipment. We need a clean microscope slide. We need a Bunsen burner. We also need immersion oil. We need an inoculation loop or wire loop, then distilled water, then compound microscope, then the bacterial sample. Then the following reagents are required for gram staining. We need crystal violet, we need grams iodine, then ethyl alcohol, as well as safranin for the staining of the bacterial cells. Now, what's the procedure for the gram staining technique? The process involves the following steps. One, using a sterile wire loop, obtain a small sample of the bacteria and make a circle smear in the middle of a clean glass slide. The second step is that you allow the smear to air dry and then heat fix by passing over a Bunsen burner flame. Make sure not to touch it to overheat. Now the stained slide is first air dried and then it's now heat fixed by passing the sample over the Bunsen burner flame. That's to fix it. After that, you now place the slide on a staining rack and gently flood with the primary staining crystal violet for about 1 minute. Then you now tilt the slide and gently wash with distilled water. The fifth step is that you gently flood the slide with iodine for about 1 minute and then tilt and gently wash with distilled water. Then the sixth step is decolorize. You decolorize with ethyl alcohol or acetone for about 5 seconds. Then you rinse with distilled water and gently flood with safranin for about 1 minute. Then 8, now tilt the slide and rinse with tap water. Then the ninth step is to blot it dry to remove any excess water stain and mount on a microscope slide to observe. Take note that you will observe that gram-positive bacteria will stain violet due to the presence of a thick layer of peptidoglycan in the cell walls, which retains the crystal violets these cells are stained with, while gram-negative bacteria stain red, which is attributed to the thin peptidoglycan wall, which does not retain the crystal violet during the staining process. Thanks a lot for watching. Thanks for watching.